All right, the last section in this chapter is going to be on the central dogma, and I would argue that this is the most important section because it's going over protein synthesis. And there's going to be a lot of questions, and there's going to be a lot of kind of complexity here, but we really have to kind of dive deep into this and look at these details a little bit more in depth than you learned it in ninth and 10th grade bio. Now, the great thing is you've already been through the first step. Replication is the first step. So DNA copying itself is the first step of the central dogma. And what a dogma is just like a set of rules or set of like processes that molecular biology abides by. And this is all life. This isn't just like eukaryotic life. This is every single life form out there we see go through transcription and translation. Transcription is taking a DNA sequence and copying it into an mRNA sequence. What that's like is like a photocopy of the DNA. The translation then is taking that mRNA, it's going to leave the nucleus as it says there, and it's going to be read by a ribosome and converted into a sequence of amino acids. You can see that process here. We're going to transcribe DNA into mRNA, and that's going to be a photocopy of the DNA, and that's going to then later be translated into a polypeptide or a protein. If you can remember, this is that primary sequence of proteins of amino acids that we learned about in like chapter 3 when we talked about proteins. But we're going to be learning about this process, these next two processes, pretty in depth here going forward. All right, so let me go back here. Come on, go back. Can I go back? Oh, all right. So RNA, here's the big difference between RNA and DNA. RNA has a ribose sugar instead of a deoxyribose, and we replace thymine with uracil. In this case, you can see here this DNA is going to be adenine, guanine, cytosine, and thymine. In RNA, we're replacing that thymine here with uracil. It's very similar looking to thymine, but it's going to be again different. RNA is also single-stranded, which means again it only has one strand of the nucleotide sequence. Here's what it looks like between ribose and deoxyribose. You can see it's just an extra oxygen group here. And I know that you're thinking like, well, I always pictured ribose being vastly different than deoxyribose. No, it's very, very similar. It's only one little oxygen that's different. If I had to choose between DNA and RNA, I'd choose RNA because that has a U in it. Okay, bad joke. Sorry. All right, so a couple different types of RNA. We're going to have mRNA, tRNA, and rRNA, and a lot of students have a tough time understanding these. And they are different structures of RNA, but they have essentially the same RNA nucleotide. So you can have a nucleotide in any one of these, and it's going to be the same nucleotide. It's just the structure is different. It's like having maybe like metal. You can have metal in many different forms, and the structure is different. Maybe you have like a coat hanger. Maybe you have like a wire. Maybe you have like a twisty tie for like bread. So like all of them have metal in it, but they're just different structures of metal. mRNA is messenger RNA. This is what's copied and transferred to the ribosomes in the cytoplasm. This here is just a single strand. It's not bonded on itself at all. tRNA is called transfer RNA. This is what transfers amino acids into transfers amino acids to the ribosomes. We're going to learn about this a little bit later. And rRNA, this is what makes up the ribosomes. There's two parts to a ribosome. It's protein and rRNA. All right, I'm not going to show you this video. I'll show you in class though. But the analogy I like to give is DNA is like a book in the library. With this book, it's going to be kind of weird because this library doesn't allow the books to leave. And this book has a recipe that we want to make. And so what we're going to do is we're going to make a photocopy of that recipe. So that's called transcription. We're going from DNA to RNA, specifically mRNA. Now this photocopy will have all of the same stuff, all of the same instructions of the original DNA strand or the original book, but it's going to just be a photocopy in one sheet. What happens then is that we have this translation. So again, we're going to take that photocopy, not the original book, because that can't leave, and we're going to take that photocopy and we're going to take it home to a kitchen and we're going to make a cake. Now that taking the instruction pamphlet home, taking the instruction photocopy home, and translating it into a protein is like translating the photocopy to the cake. So the cake in this case is the protein. Now, the locations here are going to be the library is the nucleus because the book can't leave the nucleus. And the kitchen where you're making this at is the ribosome. So we have to take the instructions from the library to the kitchen to make the protein, or in this case, the cake in the analogy. All right, next up is RNA polymerase. And this is going to be an enzyme that's very similar to DNA polymerase, but it's actually going to make RNA instead of DNA. Now, the crazy thing about this is it's going to bond with this sequence called a promoter. And the promoter is just a relocation that's going to be where the RNA polymerase is going to bind to and help facilitate transcription. We're going to learn a lot more about promoters in the next chapter. But the what I was talking about, the crazy part here is it doesn't need another enzyme to unzip the DNA. It actually does both. It's going to unzip and make mRNA. So this enzyme is pretty cool because it does the kind of the job of two different enzymes. Now this also runs along the template strand in a 5 to 3 prime direction, just like the DNA polymerase. And this is really important to understand. It runs along and forms the complementary strand of DNA from the template strand, and that what it makes is called like the coding strand, and I'll explain that here in a little bit, but it's really tough to understand this terminology. Here we go. So you can see here the template strand, it's going to run along this. So again, you're going to see that it's going to run along, making the 5 to 3 prime mRNA. Now it's going to make an exact copy of the coding strand. Here's how to remember it. This strand codes for the protein. However, if we use this strand, we're going to get the opposite, which doesn't help us. We have to get the copy of this. So we have to use the template strand to make that mRNA. Again, you can see here coding versus template. We have our DNA, it splits. We use the template to make a copy of that coding strand. Now, if you look here, the copy of the coding strand doesn't have T's, it has U's. So that's why we have to use a template strand, because we want this strand up here. And then again, we're going to take that mRNA and we're going to make a protein out of it. You can see this is RNA polymerase. This is what unzips the DNA and actually also zips it back up as well. It's a pretty cool enzyme. But you can see here how it's making the, from the template strands, making mRNA. Here's another image of it. I like both of them. They're pretty cool. All right. Ooh, let me go back. So RNA polymerase is going to stop transcribing the gene when it comes to a termination site, and we're not going to talk about that just yet. We're going to help talk about that in, again, next chapter a little bit more. But after this attaches and makes the mRNA, it's going to detach and either go find another gene to transcribe, or just kind of wait around till they need specific genes. Now I'll probably show you again. This is in class a little bit. I like that video, but it's pretty intricate. It looks at molecular, like molecular imaging, and like a really small level of the enzymes involved there. You can see our 2, 3 memes that I'm going to fly through here, and I'm not going to bore you with the memes here. Okay, so the codons. These are triplet codes of nucleotides. You can see here that the nucleotides are all in a row here. And when we have 3 nucleotides in a row, that's going to be called a codon. So this is a codon, this is a codon, this is a codon. So, all right. So with these codons, they're going to be degenerate, which means that several amino acids are going to code for more than one codon. This will make more sense when we look at a codon chart next. And they're also unambiguous, which means that each codon only has one meaning. So again, AUG will only code for methionine. ACG will only code for threonine. However, threonine might have several different codons that code for that one specific amino acid. I know this might be kind of confusing. Let's look at a codon chart here in a second. Let me go back here. So we have a couple things here: start codons and stop codons. Start codon, there's only one. It's AUG. 3 stop codons: UAA, UAG, and UGA. I really probably won't ask you to remember the stop codons or the start codon. I mean, by the time you get done with this, you should know the start codon at least and have some idea what the stop codons. But we have charts for this, and it's like a periodic table. Why memorize things when you have a table? But some things just get memorized anyway. All right, so what happens is the mRNA sequence is going to leave the nucleus and it's going to find a ribosome. At that ribosome, the tRNA molecules are going to come in and they're going to attach the amino acid together. Now, this is a clover leaf RNA molecule, and you can see here we have this anticodon. We talked about codons before. These are just a 3 base sequence that's going to align with the RNA here. And you can see here GAA. It corresponds to CUU here. So it's going to bring in complementary base pairing from the codon to the anticodon here. Now, like we said, we had that codon chart. You can see here CUU will code for amino acid leucine. We don't use the anticodon. We're only using codon charts here. Here's that here's the codon chart where you can see the start and stop codons here. You can see how we have the codon, the first letter in the codon, you see U, G, A, and C. Then you go to the second codon, which is this line here, and then the third codon. So let's do one real quick. Let's do GGG. So GGG is going to code for glycine. Let's do CCC. So CCC is going to code for proline. Let's do AGU. So AGU codes for serine, or SER. Now on the outside here, these are your amino acids, and there's 20 of them. Don't memorize them. I had to do it in biochemistry in college, but it will help you understand what these letters mean. Here's another way of looking at it. Let's do just a quick one here. A, C, A, you can see it means threonine. This one gives you the actual amino acid, which some students like, but I like the last one because it's easier. This wobble hypothesis is going to talk about how the first two letters are the most specific when looking at an amino acid. So again, G, C, and then you can see here all four of these letters, the third letter in the, or the third nucleotide in the codon, it really doesn't matter. So like GCU, GCC, GCA, GG, or GCG, all code for alanine. And that's just saying the first two letters are the most specific, and just because that's how things evolved back then. Again, billions of years ago when this process evolved, it didn't need to be specific to the last letter. It just really needed to be specific to the first two. And you can see there is some, um, like, oh, okay, well, these last two here, there is a difference, but there's still a lot of variation or lack of variation at the end. So, it's just really more important that the first two get bonded than the last two. All right, so in a ribosome, you're going to have three different parts. You're going to have the A site, the P site, and the E site. So what happens at the A site is that you have the next tRNA molecule coming in with the amino acid. It's going to bond the anticodon with the codon. What happens is this or the ribosome then moves down a little bit. The last tRNA molecule in this E site gets exited out or ejected out. Then the P site, this is where the two amino acids here are going to bond together. You can see lysine and valine are going to be next. And then it keeps on moving. So the P site is where the polypeptide starts to grow and starts to bond. And the A site's where it comes in. The E site's where the tRNA molecule leaves. Now, this tRNA will go back and it will find another amino acid to bring into the ribosome when it's needed. This is a great representation, and yep, here we go. And it's in German, actually. I know it's crazy that it's in German, but this is the best representation of an animation that I've seen. You can see here the anticodon is going to bond with a codon. You can see the A, P, and E site. As this travels down, we're going to have tRNA molecules bringing in these amino acids. You can see here they're going to come down. They're going to come down more and more. And as they exit, you can see they're not bringing their amino acid along with them because that amino acid is now bonded together, creating that primary structure of the protein. When it hits the stop codon, that's when it stops producing or stops bringing in those tRNA molecules. So again, that starts at a start codon, which is AUG, and stops at one of those three stop codons.